Garrett FitzGerald is Irish and therefore European by birth, not only, but by cultural and spiritual vision. He did his fantastic academic career largely in the States, initially at Vanderbilt University in Nashville, and then during the past 25 years at University of Pennsylvania in Philadelphia, where he is currently the director of the Institute of Translational Medicine and Therapeutics. Garrett's contribution to science is fundamentally in the field of the clinical pharmacology of pain, inflammation, and thrombosis. He contributed enormously in transforming a very old drug, aspirin, from being an analgesic anti-inflammatory agent into being a life-saving drug, a drug that can protect your heart from myocardial infarction, and later demonstrating that other members of the same family of non-steroidal anti-inflammatory drugs can have the opposite effect of aspirin, that is, they can increase the risk of myocardial infarction. In the past 10 years, his contributions have been in another area, which is in part related to the previous one, which is the area of biological clocks, particularly in the cardiovascular system, the molecular biology and genetics of clocks, and the idea that as we age, there may be a slowing of our clocks, and this slowing of our clocks may contribute to aging. Garrett, it's a great pleasure having you as a member of the Accademia dei Lincei.
So thank you, Carlo, for those very kind words. It's a great honor as well as a great pleasure for me to be accepted into the Academy and to have the opportunity to chat with you briefly today. As Carlo said, I work in two areas, Cox and clocks, and given the demographics of the membership of the Lincei, I thought I'd allude to the fact that, as we all know, time devours all things. So first of all, let's think about Cox. So arachidonic acid is a lipid constituent of cell membranes. It's subject to metabolism by two cyclooxygenase enzymes that are known colloquially as Coxes, Cox-1 and Cox-2. And they catalyze the formation of bioactive lipids called prostaglandins that activate a series of receptors that have conflicting biological roles. So why should you care about this pathway? Why should a general audience care about this? Well, amongst the most commonly consumed drugs in the world are non-steroidal anti-inflammatory drugs, and the older drugs like ibuprofen and naproxen inhibit both Cox-1 and Cox-2, and they relieve pain and inflammation mainly by suppressing prostaglandin E2 that is mainly formed, but not exclusively, by Cox-2. And as I'll allude to, insights into the structural biology of these enzymes has allowed for the development of non-steroidal anti-inflammatory drugs that target specifically Cox-2, and these were developed to minimize the likelihood of adverse gastrointestinal adverse effects. Another reason you should care about this pathway is that low-dose aspirin, as Carlo alluded to, and whose work has contributed very substantially to the development of this concept, hits predominantly Cox-1. And a major product of Cox-1 is thromboxane A2, which causes platelet activation and vasoconstriction. And the efficacy of low-dose aspirin in preventing cardiovascular disease is largely attributable, if not entirely attributable, to this property. So, of course, this is a singular biochemical pathway, and it's a gross simplification of the connections that we know amongst proteins that are relevant to this pathway. And just to give you an illustration of how the gene ontology terms fall together within this very complex and comprehensive network, we see areas that relate to arachidonic acid metabolism, highlighted here, that relate to the response to stress and inflammation, highlighted in red here, and platelet activation, highlighted in blue. But as you can see, this leaves much of the proteomic connectome uncovered, despite these being the primary actions of these drugs that target this pathway. So when these two proteins were crystallized, a surprise to some was how similar they both looked. So here we're looking at a structural overlay of Cox-1 and Cox-2. The substrate, you'll remember, is a lipid. It gains access to the active site that's buried deep within the proteins by a hydrophobic channel. And that's one of the distinguishing features between Cox-1 and Cox-2, as I'll describe to you in a moment. But why do we have two cyclooxygenases? Why do we have two COXes? And that's a question that we don't really have the answer to yet completely. But one approach that we took together with Colin Funk was to play around with the genetic structure of the two enzymes in a way that we took Cox-1 and put it under the promoter of Cox-2, and took Cox-2 and put it under the promoter of Cox-1 to see what properties were retained by the same enzymes when they were put under different regulatory control. And under stimulated conditions, we found that in the Cox-1 into Cox-2 setting, we could determine that thromboxane, as predominantly reflected by its metabolite in urine, derived mainly from Cox-1, and a counter-regulatory prostaglandin, prostacyclin, that inhibits platelet activation and relaxes blood vessels, predominantly comes from Cox-2. Well, as Carlos said, low-dose aspirin, if you have had a heart attack or if you've had a stroke, is actually quite effective in reducing the likelihood of a second event. And this is an overview analysis from workers in Oxford. They are indirect comparisons across dose, but as you can see, it looks as if perhaps lower doses of aspirin are even perhaps more effective than higher doses that would inhibit not just Cox-1, but Cox-1 and Cox-2 at the same time. Now, people think of taking aspirin as a primary prevention. In other words, people who've never had a heart attack or stroke. And the evidence for doing that is fairly weak. It does reduce the very low incidence of a heart attack, but it more or less trades that with the likelihood of a very low incidence of a serious GI bleed. And indeed, in this nice review by Carlo and his colleagues, you can see that this propensity to bleed in response to even low-dose aspirin increases as you get into the demographics that are most relevant to the membership of the Accademia della Lincei. So as I said, there are some differences between Cox-1 and Cox-2, and one is in the hydrophobic channel between the two enzymes, which is more accommodating in terms of broader substrate recognition for Cox-2, but also has this side channel that turns out to be the structural basis for being able to develop selective Cox-2 inhibitors. And we and others showed that these drugs could increase the likelihood of causing a myocardial infarction by suppressing this protective prostaglandin, prostacyclin. So in fact, in mice, if we delete the gene that is activated by prostacyclin, we increase the likelihood of response to a thrombogenic stimulus. If we use a small molecule inhibitor of Cox-2, we have the same effect. If we mimic the effect genetically of low-dose aspirin, we delay the time to thrombosis, but if we put a Cox-2 inhibitor on top of it, we accelerate it again. And if we delete Cox-2 just in the endothelial lining of blood vessels, we have the same effect. So these and other studies provided a molecular basis for why these drugs might be likely to cause serious adverse effects. And indeed, eventually, a placebo-controlled trial looking for a new indication of one of these drugs, Vioxx, showed unequivocally that compared to placebo, it actually caused myocardial infarction. And many people reacted with surprise to this. By now, there have been 10 randomized trials that have shown, placebo-controlled trials, with structurally distinct Cox-2 inhibitors that have shown that this can occur. And this is aggregate data from the Oxford Overview analysis showing that not just Vioxx, but also Celebrex has a very similar effect, as would be expected from their pharmacology. So many of you may know this book by Nassim Taleb about black swans. They don't exist unless you go to Australia. So was Vioxx a black swan? And I thought this whole idea was caught quite nicely by this letter to the Financial Times by a risk manager before the crash. He said, the point for risk managers is not that the numbers or models do not accurately forecast rare events. It is that obtaining and understanding accurate information itself is a rare event. It's not the unexpected that causes trouble. It's what is ignored, unavailable, or untrue. So I'm going to switch now from Coxes to clocks, and in the blink of an evolutionary eye, things have changed with respect to our circadian rhythms. For a very long time, humans were operating under conditions where there was a clear distinction between light and dark. There were clear thermal contrasts. We ate quickly what we killed. And now we have, in developed economies, continuous access to some form of food. We live in very controlled thermal environments. We have night lights, we have cell phones, we have tablets. And for those of us members of this distinguished Academy, there is something else that we're subjected to, and I'm a prime example of that. So on November the 5th, I was out working in a Google company in San Francisco. I flew home that night to Philadelphia to vote against Mr. Trump. On November 7th, I got on a plane again, and November 8th, here I am in Rome. So circadian disruption by travel and by shift work can do bad things to us. How do we know? Well, certainly if we look at mice and we subject them to the same type of jet lag that I'm subjected to, we can look at the circadian rhythms of core clock genes across a range of tissues, very nice oscillatory rhythms, and look what happens when you're misaligned. So if my talk is unintelligible, you can blame my misalignment. So a few quick fundamentals on the clock, which I'll skip through. There's a master clock in the brain, in the suprachiasmatic nucleus. Clocks in all peripheral tissues, except for some strange reason in the testis. We could have a symposium on that. There are diverse signals from the center that can entrain the periphery, but the periphery retains a capacity for autonomy. And environmental cues, particularly light and temperature, adjust us to a 24-hour rhythm every day. And it's an anticipatory system. It gets us ready for what's coming. But why should you care about clocks? This is a general audience. Most of you don't work on clocks. I'll give you a couple of reasons as to why you should care. First of all, this is a highly conserved system. This nice slide from Joe Takahashi shows that this is conserved across a long evolutionary period. Things that are conserved are usually important. I won't get into it, but there are multiple levels of regulatory control as far as the clock is concerned, and there's much redundancy within that regulatory control. And those two things usually point to biological systems that are important. And finally, in this nice work from Eric Schadt, he looked at biological networks that tied together networks across tissues. And he isn't in the clock business and wasn't in the clock business and hadn't even noticed the fact that right in the center of these interconnected networks is the biological clock network. So there are reasons to care. So if you break the clock in mice, what you do is you break systems that rely on cross-tissue interregulation. And metabolism is a beautiful example of that. And this is just showing the cell-selective breakage of the clock across a range of tissues that recapitulate elements of what we call the metabolic syndrome that is a scourge of contemporary society. And I don't have much time to go into it, but it's also very relevant to immunological integration and the propensity to inflammatory response. So here, just to illustrate that, is the fact that the lethality of administering a dose of LPS to mice is highly dependent on the time at which you administer it. And if you break the clock in macrophages that are important cells with respect to inflammation, you abolish this time dependence in terms of the propensity to lethality from LPS. And of course, as human beings, we know there is time-dependent expression of many disease phenotypes and indeed also of many of our physiological functions. So we're very interested in aging, as Carlo alluded to, but I'm not going to talk about that today. But we're also interested in trying to get at the human biology of the clock. In that regard, we want to, in a study that we have just started, to try and get our hands on oscillatory rhythms in multi-omics data, as well as in remote sensing data in free-ranging humans. So given that our intra-individual variability of behavior creates a huge amount of noise, can we detect oscillatory rhythms despite that noise that might be replicable across individuals? And this slide just illustrates the amount of noise in a pilot study, and yet the major contribution of variance of time rather than intra-individual variability in a range of multi-omics data. So that begins to let us put together proteins, transcripts, metabolites, epigenomic signatures to look for time-dependent oscillation of networks. And this is just an example of one of them, which is inflammatory networks detectable by multi-omics analysis in plasma. So I'm finished, almost on time, and I've just finished a wonderful book that I'd recommend to all of you, which is an homage to Don Giovanni by Murakami, which is called Killing the Commendatore. And it comes back to this Tempus, Edax, Rerum. Time devours all things. So I'll end by just reading this sentence from it. But it went without saying that his life was bounded by time, space, and probability, like everyone else's in this world. None of us could escape these constraints as long as we lived. Each of us was enclosed by sturdy walls that stretched high in the air, surrounding us on all sides. But, he says, probably, so maybe he heard the talk that preceded mine and knows that there's a lot yet to understand. Thank you.